most people believe they would know if something was wrong inside their body. They trust that pain is the warning system, that symptoms are the signal, that if something serious were happening they would feel it. And that belief, that deeply human, completely understandable belief, is precisely what makes silent chronic inflammation so dangerous. Because it does not announce itself. It does not send a signal you can feel. It does not show up as swelling or fever or discomfort that would send you to your doctor's office. It runs silently, invisibly, at a level far below any symptom you would ever notice, deep inside the architecture of your cells. And while it runs, it is doing something that most doctors never explain to their patients, something that changes the biology of aging at the most fundamental level possible. Chronic low-grade inflammation does not just damage tissue over time. It breaks DNA strands directly. It overwhelms the molecular machinery your body uses to repair those breaks. And it creates the precise internal environment where cellular errors stop being corrected and start accumulating, the environment where serious disease finds its footing and begins to grow. But here is what I need you to hear, because this is the part that changes everything. One of the most powerful tools for interrupting this process does not come from a pharmacy. It does not require a prescription. It does not cost hundreds of dollars a month. It is sitting in your kitchen right now. You have almost certainly cooked with it hundreds of times in your life. And almost nobody is using it in the way the research actually shows it needs to be used to produce its full biological effect. Today I am going to show you exactly what it is, exactly how it works at the cellular level, and the specific preparation mistake that causes most people to get essentially zero therapeutic benefit from it even when they think they are using it correctly. But before I show you the food, I need to make sure you understand something about what inflammation is actually doing inside your body. Because once you see this clearly, you will never think about what you eat the same way again. My name is Dr. Katherine. I have spent years in clinical practice and research studying the biology of aging, inflammation, and what separates patients who maintain genuine health resilience into their later decades from those who do not. And one pattern that kept appearing in the research, in my patients, in the data I reviewed across institutions and populations, kept pointing back to something remarkably simple. Not a complex intervention. Not an expensive protocol. A food. One specific food, prepared in a specific way, that has been shown in controlled clinical research to simultaneously suppress the molecular pathways driving chronic inflammation and activate the genetic programs your cells use to repair damaged DNA. Before I tell you what it is, leave a comment below and let me know where in the world you are watching from today. I read every single one, and this community genuinely means everything to me. Let me walk you through the biology first. Because understanding the mechanism here is what makes this information genuinely actionable rather than just interesting. Every single day, your DNA takes damage. This is not disease. This is not something going wrong. This is simply the biological cost of being alive. Every time a cell divides, every time your body is exposed to ultraviolet radiation, environmental pollutants, or even the natural metabolic byproducts of your own cellular processes, your DNA sustains what scientists call lesions, breaks, alterations, corruptions in the sequence of genetic instructions that your cells depend on to function correctly. Under normal, healthy conditions, your body handles this with extraordinary precision. You have a sophisticated network of repair enzymes, proteins with specific jobs, working continuously, scanning the genome for damage and correcting it before errors can compound and propagate. Think of these repair systems as a round-the-clock maintenance crew operating inside every one of your cells. Their job is to patrol the genetic material, identify the sections that have been damaged, and fix them before the next cell division locks those errors into a permanent copy. When this crew is functioning well and is adequately resourced, the system works remarkably effectively. Damage is found, repaired, and your genetic integrity is maintained. Now here is where chronic inflammation enters the picture, and here is why it is so much more dangerous than most people realize. When inflammation is chronically elevated even at the low, subclinical level that produces no noticeable symptoms, it floods your cells with a class of highly reactive molecules called reactive oxygen species. These molecules are chemically unstable, and they interact destructively with the structures around them, including your DNA. Think of them as sparks landing continuously on dry paper. Each individual spark may be small, but when sparks are landing constantly, faster than they can be extinguished, the damage accumulates. The repair crew that would normally keep up with normal daily DNA damage is now overwhelmed. It is working at full capacity just to address the damage from the reactive oxygen species, and the normal background damage that it would otherwise catch and correct starts slipping through. Over time, the errors that are not caught accumulate. Mutations develop in cells that would otherwise have been repaired. And the internal environment chronically inflamed, oxidatively stressed, with a repair system running behind rather than ahead becomes one where abnormal cells are not eliminated the way they should be. This is not a theoretical model. This is the established mechanism by which chronic inflammation contributes to accelerated biological aging, cellular dysfunction, and serious disease. Inflammation and DNA damage are not two separate problems. They are one interconnected system running in a destructive loop. So the question that matters most is not simply how do I lower inflammation. The question is how do I lower inflammation and simultaneously give my DNA repair systems what they need to catch up and stay ahead? That dual requirement, anti-inflammatory action and DNA repair activation happening at the same time, is precisely what the food I am about to describe delivers. And it delivers it through mechanisms that have now been measured and documented in peer-reviewed research using the same laboratory methods applied to pharmaceutical drug evaluation. The food is garlic. And before that word lands too simply and you move on with the assumption that you already know this, you do not know this. Not the way the research actually describes it. Because the garlic most people use, prepared the way most recipes instruct, does almost nothing therapeutically. The garlic used correctly is a completely different biological event inside your body. Garlic contains a family of organosulfur compounds, sulfur-containing molecules that, when activated appropriately, produce effects at the cellular level that are measurable, reproducible, and clinically significant. 3 specific mechanisms are worth understanding in detail, because each one addresses a different part of the inflammation and DNA damage loop. The first mechanism involves a compound called allicin. When a garlic clove is physically crushed or chopped, an enzyme called alliinase is released from the broken cell walls. This enzyme immediately initiates a chemical reaction, converting a precursor compound into allicin, the molecule responsible for garlic's characteristic sharp aroma and the majority of its biological activity. Allicin does something specific and measurable once it enters your system. It blocks a molecular pathway called NFKB, the master regulatory switch for inflammatory gene expression in the human body. NFKB, when it is chronically activated, which research suggests is the case in the majority of adults past midlife, keeps the immune system in a state of continuous low-level alert. It sustains the production of pro-inflammatory signaling molecules even in the absence of any actual threat. It maintains the precise internal chemical environment in which DNA damage accelerates and the repair enzymes are perpetually overwhelmed. Allicin, by blocking NFKB activation, interrupts this cycle at its source. Not as a one-time event, but consistently, meal after meal, day after day, and that consistency, as we will discuss, is what makes the difference between a food and a therapeutic practice. The second mechanism involves a different organosulfur compound called S-salicystine, or SAC. SAC is found in garlic in its fresh form but is particularly concentrated in aged garlic extract, garlic that has been processed through a specific aging method that preserves and concentrates certain bioactive compounds. SAC activates a different molecular pathway called NRF2, and NRF2 is the regulatory system that functions as what I think of as the foreman of your DNA repair crew. When NRF2 is switched on, it sends signals throughout the cell to upregulate the production of antioxidant defense enzymes and, critically, to increase the output of the specific proteins responsible for identifying and repairing DNA strand breaks. This includes enzymes like OGG1, which repairs one of the most common and consequential forms of oxidative DNA damage. By activating NRF2, SAC is not just lowering the amount of damage reaching your DNA. It is increasing your body's capacity to find and fix the damage that does get through. This is the direct molecular link between garlic consumption and DNA repair activation. Not garlic making you feel better in some vague sense, garlic sending a precise biochemical signal to your genome telling it to defend itself more aggressively and repair itself more efficiently. The third mechanism involves a compound called diallyl sulfide. This organosulfur molecule directly reduces the production of two specific inflammatory signaling proteins, interleukin-6, known as IL-6, and tumor necrosis factor alpha, known as TNF-alpha. These are the two most closely tracked inflammatory markers in clinical medicine. When IL-6 and TNF-alpha are chronically elevated, they suppress immune surveillance, the body's ability to identify and eliminate abnormal cells, and they directly impair the function of DNA repair enzymes. By reducing these two signals, diallyl sulfide removes one of the primary reasons your repair systems cannot keep up. It is clearing the interference that was slowing down the maintenance crew, so they can return to doing their job at full capacity. Now let us look at what the research actually shows, because the science here is not preliminary and it is not from a single institution or a single study design. Researchers studying adults with elevated C-reactive protein, the standard clinical biomarker for systemic chronic inflammation, found that regular daily consumption of raw garlic over a 12-week period produced a measurable, statistically significant reduction in CRP levels. The magnitude of that reduction was comparable to the effect produced by low-dose anti-inflammatory medication with no pharmaceutical side effects and no prescription required. The anti-inflammatory effect of correctly prepared garlic was not subtle. It was clinically meaningful. A separate body of research examining the relationship between organosulfur compound intake and the frequency of DNA strand breaks used a laboratory technique called the COMET assay, which measures the structural integrity of individual DNA strands directly. Participants who consumed garlic regularly showed significantly fewer DNA breaks per cell compared to control participants who did not. The difference was measurable after just 4 weeks of consistent consumption. This was not a change in how people felt. It was a change visible under laboratory analysis in the physical structure of their genetic material. And research specifically examining aged garlic extract and its SAC content found that consistent daily intake equivalent to approximately 2 to 3 cloves of fresh garlic produced measurable upregulation of OGG1, one of the body's most important DNA repair enzymes, within 6 weeks. The repair system was not just maintained. It was strengthened. The crew did not just keep working. They were given more resources and became more capable. So how do you actually use this information? Because science that does not translate into daily practice serves no one. The dose that the majority of clinical trials producing measurable anti-inflammatory effects used is 2 to 3 raw garlic cloves per day. 1 clove produces insufficient allicin and SAC to reach therapeutic effect. 4 or more cloves does not meaningfully increase the benefit and may produce digestive discomfort in some people. 2 to 3 cloves, daily, consistently, that is the target. The preparation method is not optional. It is the difference between garlic functioning as flavoring and garlic functioning as a genuine therapeutic food. Crush or chop the garlic. Never use it whole. Allicin only forms when the cell walls of the garlic clove are physically broken. The alliinase enzyme and its substrate are stored in separate compartments within the cell, and they only make contact and begin the reaction when those compartments are disrupted by crushing or chopping. A whole clove passing through your digestive system produces essentially no allicin and delivers none of the primary anti-inflammatory benefit. The mechanical step is not a preference. It is a biochemical requirement. Now here is the step that almost no one follows, and it is the most important single thing I will tell you today. After you crush or chop the garlic, wait 10 minutes before cooking it or eating it. Allicin requires approximately 10 minutes after the enzyme reaction begins to fully form. If you take freshly chopped garlic and immediately drop it into a hot pan, which is what virtually every recipe instructs you to do, and what most people do every time they cook with garlic, you are applying heat to an enzyme that has not yet completed its work. High heat denatures alliinase almost instantly. If the enzyme is deactivated before allicin has had time to fully develop, the conversion reaction stops. You get the flavor. You get the aroma. You get none of the medicine. Crush or chop your garlic, set it aside on your cutting board, and wait 10 minutes before it touches heat. That 10-minute window is the entire difference between using garlic as a condiment and using it as a clinically active food. This single change, this one adjustment to the timing of a preparation step, is what separates the people who eat garlic their whole lives and wonder why it doesn't seem to do much from the people who are actually giving their bodies access to everything the research shows garlic can do. The best time to consume garlic is with your largest meal of the day. The organosulfur compounds in garlic are fat-soluble, meaning their absorption is significantly enhanced in the presence of dietary fat. Adding your crushed, rested garlic to food that contains olive oil, avocado, fatty fish, or another quality fat source improves the bioavailability of the compounds most responsible for its anti-inflammatory and DNA protective effects. Raw garlic added at the end of cooking after the heat has been turned off or reduced significantly preserves more of the allicin than garlic that has been sautéed from the beginning of preparation. If raw garlic is not tolerable for you due to digestive sensitivity or taste preference, aged garlic extract in supplement form is the only processed form of garlic shown in research to preserve the SAC content responsible for NRF2 activation and DNA repair enzyme upregulation. Standard garlic powder, garlic salt, and most commercial garlic preparations do not preserve the relevant compounds in therapeutically meaningful concentrations. If you choose a supplement, look specifically for products standardized to SAC content and produced through an aging process validated in published research. I want to close with something that I think is the most important framing for everything we have discussed today. The inflammation running silently through your body right now is not something your body has surrendered to. Your repair systems have not given up. The enzymes responsible for scanning your genome and correcting daily DNA damage are still working. The antioxidant defenses your cells were designed to deploy are still present. The NRF2 pathway that, when activated, tells your body to defend its genetic material more aggressively, it is still there, waiting for the right signal. Your body does not need a pharmaceutical trigger to activate these systems. It needs the right nutritional information. 2 to 3 crushed cloves. 10 minutes of waiting. Every day. That is the practice. Remarkably small for what it initiates at the cellular level. Remarkably accessible for what the research shows it can do over weeks and months of consistent application. Chronic inflammation does not announce itself with fanfare. It works quietly, steadily, accumulating damage below the threshold of any symptom you would notice until one day the consequences become impossible to ignore. But you do not have to wait for that day. You can start giving your body the signal it needs to push back, starting with your very next meal. I am dear Katherine. If this video gave you something genuinely useful today, something you will act on, something that changes how you approach your kitchen and your health, please hit the like button right now and subscribe to this channel. Every week I bring you honest, science-backed health information that goes deeper than what most people ever hear in a routine medical appointment. Leave a comment below and tell me where in the world you are watching from. I read every single one. And if someone in your life is managing chronic inflammation, cognitive concerns, or simply wants to understand what their food is actually doing inside their body, share this video with them. It may be the most useful thing they see this week. Take care of yourself. Stay curious. I will see you in the next one.